So I would request your utmost attention in this first slide. So this is about the B cell development and Barsa development, which goes hand in hand. And to understand the pathogenesis of Gumboro virus or IBD virus, it is important that we understand the B cell diversity, its maturation, and the phases of Barsa development. So basically, if you see that chick during its embryonic stage, which is about, if I can mark it here, it's about 0 to 21 days. The growth, we'll first describe these phases of development of Barsa, then it will be easy for us to capture the B cell development. Basically, the growth of Barsa can be divided into 3 different phases. The growth phase, which is during the first 3 weeks, the plateau phase, where the growth retards and it has basically no growth at this point of time. That is a period between 4 to 8 weeks of age. And after 8 weeks, there is a slow progression of atrophy, that is called regression phase, and it continues till sexual maturity from 9 weeks onwards. And that is why you can see over here that during the embryonic, this ED stands for embryonic development. So embryonic development of 0 to 21 days, you don't see any Barsa, just you see the Barsa development from 0 to 21 days, where the growth is extremely, the growth rate is extremely high, and it starts retarding from 9 weeks onwards and can last till 4 to 6 months. That is up to the sexual maturity. And by the time the Barsa completely regresses. Now, this is the Barsa development grossly, but what happens inside is the B cell development. It's a period of diversity, maturation, and we'll discuss about this now. So this starts, basically Barsa development starts after the chicks is hatching out. Chicks are hatching out, but if you see the development of B cells, it starts even before that, that is the period of embryonic development. So during 0 to 8 days of embryonic development, there are formation of B cell progenitor cells. So B cell progenitor cells are precursor B cells. So basically we can divide this B cell development again into pre-Barsal, Barsal, and post-Barsal period. Now this pre-Barsal period from hematopoietic stem cells, which are non-committed stem cells, precursor B cells are formed, and they are called B cell progenitor. Now they goes into enters the Barsa during period of that is called late embryonic development phase, that is between 8 to 14 days of age. And basically these B cells are having the receptor plasticity are not matured, and basically the chicken host, the chicken immune system, they test the B cell receptor and test the fitness of the B cells during this phase. And noticeably, the B cell that we find during this phase, when the B cell enters enters the Barsa during these 8 to 14 days of age of embryonic development, is IgM type. It is IgM secreting B cells. And those are immature. Why we are calling it immature? Because the immune system, they test these B cells for their fitness, just like our defense. They test the fitness of the military personnel. So 95% of the B cells are destroyed during this event. And this is a very, very serious event, because this is going to be a strong foundation for the future immune system. This happens even in the thymus for the T cells, but it's not something relevant with the Gumboro, so we won't talk about thymus here. But here we talk about B cells, which are producing immunoglobulins. And in the Barsa, during the embryonic development phase, late embryonic development phase rather, the Barsa conducts series of tests for the B cells. I'm not going into much deeper what tests they conduct, but this is a routine procedure of the immune system during the development, that they test the B cells and declare them to be fit. And during this process, about 95% of the B cells which had migrated or maybe in the state of replication inside the bursa are destroyed. Only 5%, roughly about 5% of the B cells are declared fit, and they are still of IgM type. Now, this is a point where the B cells now become mature B cells, which is about a period of embryonic development phase of 14 to up to 8 weeks of age. Broadly, this happens. It continues thereafter, but majority of the process completes within the 8 week of age. And initially, you can see I've written ED 14 to 8 day. Initial 8 day, I mean, from ED 14, after the chicks are hatched up till the chicks are of 8 days of age, this event of maturation continues very, very vigorously. Could you just confirm that you are able to hear my voice, because my pods got disconnected?
Yes.
Yeah. Thank you. So, so this event is very, very important, because that is going to have a tremendous impact in the later stages during the Gumboro challenge and whatever interventions we are doing. So ED 14 to 8 days of age, the maturation of B cells, the process of maturation is extremely, extremely vigorous. And there is one more event. What? Let me describe this later. But what goes in the process of maturation of B cells? So by maturation, we mean the B cells diversity. And what do you mean by B cell diversity? In very short, the B cell are secreting immunoglobulins. Now, body encounters any living organism. They encounters like avians. They encounters plethora of pathogens, various kind of antigens. So the say the IgM type immunoglobulin, which is the predominant immunoglobulin that we see in the B cells during the immature stage, and as well as in the first phase or second phase of maturation. So these different antigens have a different amino acid profile, while the immunoglobulins basically structurally they are same. They have, you see this Y-shaped structure of immunoglobulin, there is a portion called variable region, and this is called Fab portion, this Y-shaped structure. And you might be having basic knowledge, because I assume that most of you are who have joined this event are veterinarians. So some of you are budding veterinarians, so you might have completed the course of immunology. So the memories are strong, and I'm kind of refreshing that this Fab portion has a binding site that has variable amino acid chains. So those variable amino acid chains, they keep on varying. So the variation will be happening according to the antigen they come across. So for say Newcastle disease, the IgM type B cells will be changing the variable region amino acid sequence so that it exactly fit for the epitopes of the Newcastle disease virus. For Gumboro, it is they change it in according to the amino acid profile of the Gumboro epitopes. For example, XYZ, there are many such antigens they come across. So for this, the B cells need to do diversification of these immunoglobulins, basically the variable amino region of the immunoglobulins, which are binding the epitopes of the virus or bacteria, the pathogens for that matter. So this diversification is basically a process of gene conversion. It's called horizontal recombination. And this gene conversion, this you can see it's written as gene conversion. It's a horizontal recombination process. It's a kind of complicated process, but as of now, it will be it will be quite okay if you just, remember this word of gene conversion or horizontal recombination. And by this way, the immunoglobulins are getting ready to counteract with different pathogens and different antigens. So this is a very, very important step. And this is happening from chicks hatching out till 8 week of age. And the first 8th day, this is happening very, very fast, right? And it will continue to happen slowly till bursa regresses. But the first ED14 to 8 day of age is extremely fast and continue at decent pace during first 8 week. In tandem, suppose the Newcastle disease, the immunoglobulin M, IgM type is ready. Now what the immune system will do, it will make a clone of those IgM type secreting B cells, which are ready to produce effective and fit immunoglobulins, which can take care of Newcastle disease virus. And once the clones are being made, they will rapidly increase their, population depending on the second exposure of that particular antigen. So these activities are also very important. Creation of antibody repertoire. So they will make stocks and inventory of those different clones of immunoglobulins, right? And then by about first week of age, immediately the process starts, by the way, after the hatch. But by about first week of age, the migration of some of these B cells happen to the secondary lymphoid organ, that is called SLO. So secondary lymphoid organs will be a site where this process that I just discussed, that IG diversification, that is by which they make different immunoglobulins against different types of antigens. The process will continue, but here the process of diversification is mediated by SHM, that is called somatic hypermutation. These are two important mechanisms, that is gene conversion, that is horizontal recombination, and somatic hypermutation, by means of which IG diversification takes place. And they will happen in spleen and gut, that is gut associated lymphoid tissues. And this start from prior to hatch, and again in first 8 days this process is happening. So basically what we see is that in first 8 day till 8 week, this process of IG diversification, the clonal selection of the B cells and its amplification, that is increasing the repertoire of antibodies, these are happening, and they are happening in very, very high pace. In lymphoid organ, another important activity takes place, which is not taking place in the bursa, by the way, that is called B cell activation. B cell activation is a process by means of which a B cell, when encounter a pathogen, gets authentication to start secreting immunoglobulin. Now remember, when these B cells have migrated to the lymphoid organ, these B cells are ready, they are fit to secrete immunoglobulin type M. We don't see other classes of immunoglobulin, so class switching will be also happening in the secondary lymphoid organ, that is the IgM type to IgY type. In my previous webinar on Newcastle disease, I explained about class switching. It happens with repeated encounter of pathogens. Now, prior to that, the B cell has to be activated. It is just like buying a iPhone or a Samsung phone from the store. You need to activate it so that it gets registered in the company's website, and you're getting a license to start the device, and you get a warranty on that. The similar process, it's also, you can compare with the two-factor authentication that we have in the Apple devices. Many of you might be using that. So B cell activation means the B cells are ready with their resources to mount immune response, that is secreting immunoglobulins, but they need to get activated. And for this activity, they require T cells. T cells actually give them this authentication to go ahead and secrete immunoglobulin. So this happens in the secondary lymphoid organ. I can explain much more about this, and this is very interesting, but this would, again, due to paucity of time, we would leave this as it is here. So now these mature B cells will start secreting immunoglobulin, and they will, some of them will get transferred in, converted to plasma cell. What is plasma cell? Plasma cell is a secreting B cells, and they also, and some part of them also form the memory B cells. So when the second encounter takes place, these B cells will start producing IgM type immunoglobulin. Later on, class switching will take place, and they will start producing even IgY type, which is happening in the secondary lymphoid organ. And in tandem, the immune system will have some, the plasma cell converted into another mold that is called, we call it mold. It is memory cell, so that they are ready to exert another strike when there is another encounter of the virus. So memory cells are repository plasma B cells, which are able to identify the antigen faster and immediately start producing the immunoglobulins, and also direct the body system, immune system, to rapidly produce the B cells against those particular antigen. So this is the entire process, you see, that is happening. And here I want to make some interesting comments, that the avian species, if you see, they have they are separated from the mammalian species, if you consider humans, by almost say 200 to 300 million years. It comes between fish and humans, that is mammals, the avian species. But the bursa and the B cell development process grossly is same. Maybe there are molecular difference, like say you see in bone marrow we have this Ig diversification taking place, whereas in bursa they have Ig diversification. In bone marrow, the Ig diversification takes place by somatic hypermutation, that is HSM, but in this case it's just opposite, gene conversion, while in SLO it is happening in SHM. So this kind of process grossly being the same, so the bursa development has been the ideal model even for the human scientists to understand how the B cell development takes place. I think you understood now how it is happening around. So it will help to understand the disease progression better in the next slide. So Gumboro, I won't talk much about Gumboro, because you might be knowing what it does in the field. Basically, this is a non-enveloped double-stranded RNA enterovirus in the avivirinavirus genus and Viridae. And it typically affects young chickens. You might have read that it affects from 3 to 6 weeks of age, but here you have to understand it can affect even the chicks at first week itself. The susceptible period depends on the level of maternal antibodies these chickens are coming up with from the parents. The chicks in a flock, at least 30 to 40% of the chicks in a flock have maternal antibody level lower than 700 IDXX titer. Even at the first week, it may get affected. It's a very early outbreak. And the primary target organ is the bursa fabricius. Why? Because it targets the IgM plus B cells, the immature B cells. And if you remember the development phase, it is there in the bursa from around ED14 to 8 days of age. And the IgM type immunoglobulin enters the bursa somewhere around ED8. And so it becomes an ideal organ for the virus to attack, because the IgM positive immature B lymphocytes are available there. And the virus is non-enveloped, and that is why it is highly resistant to envelope acting disinfectants, harsh environmental settings, pH and temperature. It remains viable for a long period in the farm. So outbreaks are caused by the resident virus of the farm, basically, with previous history of outbreaks, or maybe the neighboring farms, because it doesn't get inactivated by envelope acting agents like, say, normal detergents or quaternary ammonium compound. They miserably fail in inactivating infectious bursal disease virus. To inactivate it, you need aldehyde compounds like glutaraldehyde or formaldehyde, which are effective in even you might require some long exposure for inactivating this virus, because it's a non-envelope virus. So there are two serotypes. Serotype 1 is basically pathogenic to chickens, and serotype 2 is not relevant for poultry, but it can, of course, cause titer spike, and sometimes even in the ELISA we get titer spike, which possibly could be the exposure of serotype 2 even, but serotype 1 is the one which is causing the disease. So it can be divided into various types depending on the antigenicity and genetic aspects, classical and variant type, and level of pathogenicity on attenuated, virulent, very virulent type. So if you see the trends of mortality, 0 to 5% mortalities of we see by the variant strain, 20% mortality by the classical strains, and greater than 50% mortality by the very virulent types. Now they are all same serotype. Touchwood and God's grace, the nature's grace, I would say, it's single serotype. Now, one point I'd like to mention here is that of late, the very virulent IBDV strains, they are also known to affect the mature B cells. The mature B cells. Till now, you have seen that I was talking about immature IgM positive B cells, which is there in the system for first 3 weeks, and in the bursa before the chicks are hatched out. So that is why the chicks of 21 days, up to 21 days, are extremely susceptible. But VVIDV also known to affect mature B cells and even some IgY positive cells. But this is again not the cell of first priority. It has been studied. It is always the immature IgM plus B cells which are extremely susceptible to Gumboro infections of all pathotypes. So basically this virus, already mentioned, it's a double-stranded RNA virus. It has segmented genome, that is 2 segments. And here 2 important proteins coded by the segment A is VP2 and VP3. They are important because from the point of view of vaccine preparation or targeting the epitopes. So they are the immunodominant epitopes. So antibodies produced against these 2 epitopes are going to neutralize the virus in case of humoral immunity. So inactivated vaccine targets, even the live vaccines also targets these 2 immunodominant epitopes, VP2 and VP3. And otherwise the VP4 and VP5, they are kind of non-structural proteins because they are not on the surface structure. And VP1 is the RNA polymerase. So for our understanding, we need to remember that VP2 and VP3 encoded by the segment A of this virus are important for our strategy to produce the vaccines. So what do we understand from the first part of the presentation is that the IgM positive cells or immature B cells in the bursa follicle are extremely susceptible to Gumboro virus, and B cell activation and class switching, which is happening till 8 weeks, and it also gets augmented by the vaccines. If we do vaccine, the class switching will be much faster. It's happened in the secondary lymphoid organ, that is spleen and gut associated lymphoid tissue in case of chicken. After migration, and this migration takes place between day-old chicks to 8 days of age. So this is a very critical period. Sometimes we do vaccine during this period, so it might interact with this process. That's why this is very important to understand. Doing Gumboro vaccine in first 8 days of age, and if there is no maternal antibody, it might interact with this process, resulting in lesser IgM positive B cells and more IgY positive B cells. So this class switching would ensure that we have lesser IgM population in the bursa, and gradually it is switching to IgY, so it makes the bird lesser susceptible. So this explains why the chicks up to 6 weeks are highly susceptible, right? Because, and from 3 weeks to 6 weeks, because in first 3 weeks we see most of these B cells are immature IgM positive, and if we can augment the process of class switching and we get more IgY positive B cells, the susceptibility decreases. So after 6 weeks, susceptibility decreases rapidly, and because of more IgY positive B cells. Although IBDV is known to have susceptibility towards IgY positive B cells, but by that time also we develop immunity done by the vaccines, which we'll be discussing in the later section. So the outbreaks are primarily caused by the resident IBDVD of the farm with previous history of outbreaks besides newer challenge. So this is very important, that if you get a previous farm with a previous history of outbreak, those viruses are likely to be there in the farm, if not proper biosecurity measures are not done. So if you have not taken care of that, you are likely to get an outbreak, especially with low MDA chicks. So single serotype of IBDV, multiple pathotypes. So this is a good thing that we know that there are single serotype, so it becomes easy for the vaccine to work. And VP2 and VP3 are important immunodominant epitopes for protection. So this is very important from vaccination point of view, VP2 and VP3. So what happens during our IBDV challenge? So there could be two cases. There could be a challenge in a flock with low maternal antibody. Maternal antibodies are produced, basically they are IgY type of immunoglobulins in the parents, and they, due to vaccination in the parents, and they are stored in the yolk, which get resorbed, and they are released in the blood circulation. So 500 to 2500 IDXS titer are considered to be low MDA level, which may cause early outbreak, and higher maternal antibody, it is above 2500 IDXS, which are known to cause late outbreak, that is greater than 21 days of age. Now, in case of layers, you can see a footnote over here that up to 5 to 6 weeks of age, we can see an outbreak in layer and parents. If it is more in the broiler chickens, because layer and parents have the IgY type have higher half-life, so they last longer, and you don't see an early outbreak easily until and unless the maternal antibody levels are miserable. So normally late outbreaks are seen. So early outbreak is uncommon in case of layer and parents. It's common in broiler chickens. So we will take an average maternal antibody level somewhere around, say, 1500. And we use this as a model to understand what happens during IBDV challenge. So here is a chick with a 1500 MDA level, IDXS titer. It drops in exponentially depending on the half-life it has. If it is a layer, it may be up to 14-15 days. If it is a broiler, it will drop by 12 days. So in this case, the MDA level drops below 500, which is a critical point in terms of vaccine as well as a field challenge, that the MDA level falls below 500, it's become easy for any pathotype of Gumboro virus to cause infection. Even this for some vaccines, this MDA level is called a breakthrough titer. We'll again discuss this in the later section. So chicks will be protected for first 6 to 7 days of age, because it's dropping below 500 by this age. And we are considering in the farm, there is a continuous exposure. So the field exposure is continuous in the farm. Maybe say there is a previous history of outbreak, and there will be an IBD infection as soon as the maternal antibody, drops, because nothing is there to protect. Some but integrators, they do vaccine in the first 5 to 6 days of age by an intermediate strain or a standard strain. It's a weak, fragile strain, by the way, and they do not have much capacity to override the MDA, and they do not produce stress on the bursa. So this explains why they do that. So technically they are correct, but it might have some implications. Remember, first few weeks of their life. So what are the vaccines available? There are two different kind of vaccines. One is hatchery-activated vaccines. Another is live attenuated vaccine. That is conventional live vaccines that we have in the field. Hatchery-activated vaccines are of two types again. Immune complex type. There is a separate type that has been launched that is called MB1, and this is again not comes under immune complex category, but it's again can be given in the hatchery in day-old chicks. And a vectored vaccine, which is basically a herpes virus of turkey vectoring the PP2 protein of Gumboro. And what is immune complex vaccine? Basically, this is the IBDV virus, which is coated with the IgY, anti-IBDV IgY. And this coating is done with certain ratio of antigen to antibody. And this complex is called antigen antibody complex or immune complex. Now, in this slide, we are discussing about the advantage and disadvantage of these vaccines. So live vaccines are the conventional vaccines that we use in drinking water or eye drop route. It has early protection when you apply, but again, this application is really questionable because it challenges the consistency by the farmers who is applying the vaccine. And again, there is an interference of the maternal derived antibodies with these vaccines. To rule out the interference of maternal derived antibodies, there are development of other categories of vaccine, that is immune complex and vector. Immune complex advantage, again, it can rule out the interference by actually the maternal antibody or MDA, and they are meant for hatchery application. But at the same time, the main disadvantage is of the delayed onset of immunity. Now, this delay is due to the coating of the IgY that is done on the IBDV virus during the process of complexing. And this basically is formulation dependent, and the one manufacturer might have a better release of the virus in the bursa, thereby reducing the delay. Another might have greater delay due to different ratio of antigen and antibody used for complexing the formulation. In case of vector vaccine, it also rules out the interference of MDA, but it expresses only the VP2 protein. It misses out on the VP3 protein, and the vector itself has potential disadvantage, that is the herpes virus of turkey. And due to the herpes virus of turkey and its slow replication profile, the onset of immunity is extremely delayed. And this is something very important because it takes about 4 weeks of time for the herpes virus of turkey to elicit proper humoral immune response. The cellular immune response are also delayed by the herpes virus of turkey in accordance. So what are the potential problems with MDA? Now, suppose we have chick with a 5,000 IDXS titer, it drops by day 20, and you are doing a vaccine at around day 12, it is going to neutralize the vaccine virus. And that may cause clinical infection, poor immune response, or subclinical IBD. And how to correct this MDA interference? We do it by collecting blood at day 4, probably around 15 samples per 1,000 birds. Then we do IBD ELISA, then use deventer formula to calculate the right age of vaccinating the flock. Before I go to the slides, Dr. Vivek, I'd like to request you the next time, if I'm again getting disconnected, please do call me, because I don't have this number saved. I thought somebody is calling me, many customers, so I that switched off the mobile, right? So you give me immediately a call so that I understand that from this point I'm lost.
Yes, okay.
So yeah. The vaccination, the formula is calculating the vaccination age, the log2 titer bird percentage minus log2 breakthrough into t-half plus age at sampling and correction factor. Here, the critical part is the bird percentage, the ELISA titer of the bird representing a certain percentage of the flock. Which percentage of the, titer to consider that this would be a representative of the entire flock. So this becomes challenge. And here is the limitation based on this challenge. Suppose you have a flock having a titer of 5,000 and a bird having titer of 1,500. So one drops at day 20, one drops at day 12. And the deventer formula give you an age of 14, say. So at this age, you might be protecting a flock, birds, which in the flock, which is coming in between 3,000 to 2,500, or say 4,000 to 2,500 MDA level. But there are two extremes. One is having 1,500, one is having 5,000. So the birds falling under 1,500 or above 5,000, they are not going to be protected. There could be an early outbreak here in this early infection in here, and there could be a late infection in case of these birds. But in broiler flocks, sometimes we do not get, because the birds having good titer might be protecting the birds, the other birds in two extremes from spreading the virus. But still, in case of layers or parents, we may encounter this, because these birds are kept for a longer period of time, and they are not mixing together like the broiler chickens. So deventer formula can predict the right age accurately till MDA level or MDA CV percentage is lesser than 50. The variation of the titer is lesser than 50. So if it is moving above than that, the predictions are inaccurate and may cause potential outbreak. So the solution to this varying MDA level above 60% CV could be the immune complex vaccines or the vector vaccines. Now, I already mentioned about the immune complex, that it has got the disadvantage of delayed onset of immunity, and the vector vaccines are again, due to herpes virus of turkey, there is slow replication of herpes virus of turkey. It also elicits delayed onset of immunity. There is another class of vaccine, that is MB1, which is again a hatchery vaccine, but it rules it also rules out the interference of the MDA, but it actually solves the delayed onset of immunity issues by immune complex and vector vaccines. So in this webinar, I have taken MB1 as an example to make you understand how this advanced art of vaccines work. Say this is called evolution of Gumboro vaccines for day of hatch chicks. It is basically given through subcutaneous route, but it's a live vaccine. It can be given in day-old broilers, layers, and breeder chicks. There are many presentations and doses available. The main characteristics of this vaccine is that it has early onset of immunity. That I told that it addresses the delayed onset of immunity by immune complex as well as a vector vaccine. the vaccine virus release in the bursa is engineered in a way that it is happening individually for individual chicks, and as per requirement. For 500 bird chicks, if it is really the MDA drops up around 7-8 days of age, the virus gets released within next 24 hours in the bursa. So it is protecting at the level of individual chicks, and that is why it ensures uniform immunity, and it spreads quickly in flock, creating uniform immunity. A single dose can protect the broiler flock, so no need of booster immunization. It's a broad spectrum protection, so protects against any form of IBD virus, variant and classic. It has hatchery applications, so both in vivo and subcutaneous hatchery provide full control on the vaccination process and efficacy. And it is compatible with the inactivated vaccines available. There is no interference or with the immune response to other vaccines that can be applied simultaneously or individually. And it's successfully compared with other IBD hatchery vaccines. So the mode of actions is as follows. So if you do vaccination with MB1, either in the embryonic stage, in ovo, or in the chicks at day 1, what will happen? The virus will form complex with the MDA inside the chick. So that is the difference with the immune complex. Immune complex comes with preformed complex with antigen antibody complex. Here the virus is native. You give it in subcutaneous route, the MDA level that is available in the chick will actually coat it. And instead of neutralization, this will preserve it for a period till the MDA level of the chick gets waned off. So this is the newer technology that has been formulated by the manufacturer, so that the IGY is, instead of opsonizing the virus and clearing it from the system, it will preserve it. And it is thought that follicular dendritic cells are involved because it is given through subcutaneous route. So vaccine is released within 2 to 3 weeks. So it is happening according to individual chicks. So a chick having 500 MDA, it will release accordingly. A chick having 800 MDA will be released accordingly. So, and after releasing, active immunity is produced after replication in the bursa. It takes about 10 to 14 days time for the human immune response to kick in. It kicks in. The titer to be visibly appearing in the ELISA. And for low MDA chicks, it will be early activation of immunity. For high MDA chicks, it will be late activation of immunity. So there is a video to make you understand about this.
Protecting your chicken flocks effectively from infectious bursal disease, or IBD, is critical for your poultry operations. This highly contagious viral disease that typically affects young chickens can lead to severe losses from direct mortality, immunosuppression, and secondary infections. The challenge with field vaccination practices, when hundreds of thousands of chickens are to be vaccinated, is that they fail to adequately address the window between maternal antibody protection declining and the onset of active immunity that is provided by vaccination across the entire flock. The problem is compounded by the presence of various forms of viruses in the field, from classic and very virulent strains to variant strains, and the varying levels of maternally derived antibodies, or MDAs, in young chickens. To significantly improve protection against IBD, Phibro Animal Health is introducing the MB1 vaccine, a new approach to IBD vaccination that stimulates an early onset of immunity and adjusts the release of vaccine viruses at the correct time for each chick. The MB1 vaccine is developed from the MB live attenuated viral strain that is adapted for in ovo injection at 18.5 days of incubation or subcutaneous administration in the hatchery. It has been proven safe and highly effective in protecting against any form of the IBD virus, classic, very virulent, or variant strains in millions of birds under commercial controlled conditions. So how does the MB1 vaccine work to provide superior protection compared to other vaccines? Maternally derived immunoglobulins, or IgYs, are transmitted in a defined limited number from the hen to the offspring through the yolk sac and won't multiply or be self-produced until the chick's immune system matures. While IgYs are particularly effective in preventing IBD infections during the first days of age, MDAs often block the response to conventional vaccines. MDAs decay at a relatively constant rate with a half-life between 3 to 5 infection. The levels of MDAs transmitted from the hen to the chick can vary greatly across flocks. Thus, the situation becomes even more complex when there are a large number of chickens in the flock. Setting the correct time for vaccinating the flock is thus very challenging, since a window of optimum vaccination is very short, lasting only a few days, and also can occur at different times for each individual chicken. MB1, an evolution in IBD hatchery vaccination, addresses these challenges by individually adjusting the release of MB1's virus to stimulate an early onset of immunity. After injection, the MB1 vaccine viruses are coated with the chick's MVAs, which prevent the viruses from replicating too early in the young chick's bursa. The newly formed immune complex won't destroy the MB1 viral particles, but will instead keep it in a latent form until the proper time for its release. After the natural deterioration of the MDAs, the MB1 vaccine viruses are released and can replicate. This introduces an earlier immune response in every chick at a different time, depending on the chick's individual MDA level. Phibro's R&D team developed a sensitive method to detect the MB viral strain to prove that the MB1 vaccine release will successfully be adjusted to the individual MDA's titers. Field trials have shown that chicks vaccinated with the MB1 vaccine showed an earlier presence of the MB viral strain in the immune organs, as well as an earlier seroconversion in comparison to the immune complex vaccines, resulting in at least 4 days earlier onset of protection. The MB1 vaccine did not interfere with the response to the NDV VH vaccination. A single dose of the MB1 vaccine protects for the lifetime of the bird, and it can be safely applied in broiler, layer, and breeder chickens. The MB1 vaccine provides reliable and convenient IBD protection through a hatchery-controlled vaccination process. And it is now...
So, Dr. Vivek, once again, one more confirmation. Can you hear me?
Yes, Dr. Susi.
Yeah. So this was just a model of hatchery-directed vaccination, an exemplary that MB1 has, that it can rule out the interference of the maternal-derived antibody, as well as it addresses the delayed onset of immunity that we are having with other variants of hatchery-directed vaccines like immune complex and vector vaccines. So a lot of global trials have been done on this vaccine. We have done a trial in India as well. So you can refer to Poultry Science Journal on the field study of day of hatch. Since we are having paucity of time, I would request the attendees to go through these publications available. You can search in the name of Shishimukul Ray MB1. You will get in Google on MB1, how it functions, and also detailed literature of other conventional vaccines or different types of different variants of vaccines that we already discussed, like vector and immune complex. So forthcoming slides are just, I was about to describe you the trial that was done and what we did, what was the result. And it was a bit interesting, but I would like to skip this as of now because there are 2-3 important more things to be covered. But I will give you a sneak peek of the trial before that we complete this part. What are the correlates of protection for Gumboro then? So we understood the foundation of the B cell development, the bursa development, why the bursa is so susceptible organ for the Gumboro replication, and that is the IgM positive immature B cells. The process of Ig diversification and migration of the immunoglobulins to secondary lymphoid organs, and what critical role it might play if you are doing a vaccine very early, through mucosal route. So with that understanding, it is very important to understand the correlates of protection as well. In case of broilers, the maternal derived antibodies you see play a very important role in first 3 weeks of their life for protection. That we understood by now. Now, if you see the vaccination age, that is around 11 days or 12 days in case of broilers, and if you measure, start measuring the titer levels that we are getting after vaccination, you won't get to see visible range above positive control below 28 days of age. It starts from 20th day of age, the titer starts appearing, and it reaches peak by 35 days of age. So what is protecting the broilers from, say, 12 days of age, after the maternal antibody has dropped, till the flock is showing the titer response, say, by 28 days of age. So this trial that was done before also speaks about that. I'll just show you this data. If you see, this is a strain, live vaccine strain. This is about 4 days of age. This is the maternal antibody that we are getting. At 14 day, we are getting 548. Means it has started dropping below breakthrough. 21 days, it is completely susceptible. Now, the vaccine was done on 14 days of age. This is a live vaccine. 2280 strain of it's of MSD, and this is a typical immune response after doing vaccine. So it will drop to almost zero by 21 days of age with starting maternal antibody of 3,505, around 3,500. So the vaccine was done at 14 days of age, and we start seeing titer response by 29 days of age. So what is protecting the flock from 14 to 29 days of age? So that is the cellular immunity. In case of MB1, if you see, it starts with a similar maternal antibody level, it drops to 21 days of age, and it gives better immune response. This is because earlier replication. As I told you, as soon as the flock dropped to 151, most of the vaccine viruses were able to release in the bursa because it is dependent on the MDA-dependent decay. Whereas this 2280, it is not. It's a live vaccine, conventional live vaccine. In case of immune complex, the titer again was lower than the MB1, and the MDA level dropped earlier than that, and still MB1 had a higher immune response. In case of this also, you can see the MB1 had a higher immune response. It was much a delay. This was this group has to be compared with this group. It reached 10,000 titer, whereas it was around 2839. And this lower titer says that the immune complex vaccines has relatively delayed onset of immunity, which MB1 could address. But the thing that is important over here is that 21 days of age, it's almost zero, and 29 of days, you start seeing humoral immunity. So this is the correlates of protection. We have extremely important arm of immunity playing a very important role, most critical period of broiler growth. That is the cellular immunity. We often forget this. What is the importance of this cellular immunity in terms of vaccine application? It makes the timing of vaccination and entry of vaccine virus in bursa critical. It is not about, please remember, it is not about vaccinating the bird per se physically. It is about the time at which the vaccine virus, the Gumboro virus, is entering the bursa. That creates that environment of cellular immunity. And that is the time at which the chick is actually getting immunized. the release of the virus in the bursa, right? So if you are not able to achieve this, if the vaccine virus, if this is a conventional live virus and it is not able to override the MDA, suppose at day 14 you are doing some bird with still having 2000 level of titer, it is not able to override that, your virus will get neutralized, and proper amount of virus won't be able to enter the bursa, and the replication, the uptake by the bursa won't be proper cellular immunity won't be developed. The intensity of cellular immune response required to override, defend the very virulent pathogen, say. So cellular immunity in next 10 to 15 days is very important. So if you see in broiler life cycle, 35 days of protection itself is conferred by the maternal antibody and cellular immunity. Humoral immunity just comes later when it is already ready for marketing. Understood? So the humoral immune response is just an indicator of the fact that the virus was properly, or the bursa has uptake the virus. There's a proper uptake of virus by the bursa. It's just the indication of the fact. Humoral immunity here will just indicate if you have a strong humoral immune response, like we saw in MB1, 10,000 IDX titer. Definitely, it is going to protect. It will have an additional bit of protection. But what it will have, indication it has is that the cellular immune response was extremely strong. And the virus was properly taken up by the bursa. The uptake was proper. That is why you get a proper humoral response. With the cellular immunity, there is innate immune response, and there is cytokine environment created that actually helps in proper humoral immune response. So what we say from here is that, again, going back to the trial, see trial 3 between immune complex and MB1, both are hatchery adapted, given at 1 day of age. It starts with 2,500 IDX titer, drops to 120, drops to 458. It records a titer of 10,438. It records a titer of 2,839. The difference is, of course, there is difference in humoral immunity. There is a delayed onset of immunity, we understand. But there is one more important point is that the MB1 were able to elicit better and stronger cellular immune response than the immune complex. And it was earlier to do that. And that means the flock had an early protection. The flock got an early protection. For in case of, say, this 2280 and this has started just at 29 days of age, the humoral immune response. So the cellular immune response were much subdued compared to MB1. 2280 is the conventional live vaccine. But still it was able to protect. If you see the mortality report and all, considering Gumboro endemic in India, there were no fatality report. There is no mortality report in the entire trial. So this just goes on to suggest that it was the cellular immunity that played a very important role between 21 days to 29 days in protecting the flock, even greater than 29 days because it is lesser than 2,000 IDX titer. So now coming to broilers, so the maternal antibody and cellular immunity plays the most important role in protection. Humoral immunity is just an indicator. In case of layer and parents, it's the humoral immunity that has very important role. So first initial phase is the MDA, then followed by cellular immunity, and later it is added by humoral immunity. And immunity plays a very important role because class switching is important. Therefore, importance of inactivated vaccines comes into play. This classes of poultry need to cross that 8-week to 9-week susceptible barrier, whereas for broilers 5 to 6 weeks is enough. So first 5 to 6 weeks, the humoral and the cellular and maternal antibody plays the role. But for layers and parents, the extra mileage of 2 weeks or 3 weeks that is there, and considering the VV-IBDV challenges, and layer as a classes of poultry is extremely susceptible to Gumboro. The 2 to 3 week period, the extra 2 to 3 week period is supported by humoral, a combination of humoral and cellular immunity. That is why it is so important that inactivated vaccines has to be given in the layer chickens at the first week of their life, along with the live vaccines, to get to this level of protection. Hope you understood the correlates of protection, and then we come to the vaccination program. So this graph I kept, this is again from our study, Ray et al. 2021, a comparison of MB1 and immune complex. You see the maternal antibody dropped to 14 to almost 0 by 14 days of age. And from 14 days till you see 28 days, there is no proper humoral immune response. So the cellular immunity plays the role here. As soon as the virus gets released in the bursa, the cellular immune response, the innate immunity kicks in. The innate immunity followed by cellular immunity kicks in. So suggested vaccination program is in broilers. If you are doing day-old chick vaccination, we also call day of hatch vaccination with MB1 live or any immune complex vaccine through subcut route, you don't have to do any vaccination later on. This will automatically take care of the maternal antibody, the varying level of maternal antibody, and will give considerable amount of protection to the flock against odds of challenges. And in India, we have endemic areas, extremely endemic areas of Gumboro, like northeastern part of India. We have seen MB1 to be very, very effective there. And people placing the chicks, outsourcing the chicks, have much variation of MDA level because they don't have their own parents. They don't have any control on the vaccination program of their parents. So the MDA level has a lot of scatter in it, a lot of variation in it. So in those cases, this vaccine works extremely fine. But if you're going with a live vaccine, I would seriously suggest you to check by deventer formula and then place the chicks. And you can do vaccination both through drinking water or eye drop route. Just see that if you have any other eye drop vaccines in preceding 2-3 days, you can give it in drinking water. Remember, Gumboro, both drinking water and eye drop route are legitimate route of vaccination. In fact, naturally, the fecal-oral route is more primary route of infection. So drinking water is extremely legitimate. Layer chickens, day 1 to 3, if you are going with the hatchery after vaccine, in case of Gumboro, you give in the hatchery, but in broiler, you give in the hatchery. But in case of layer, you have to, you can give a vaccine in between day 1 to 3 in subcutaneous route. But day 5 to 6, you give IB inactivated vaccine. This is either Gumbin VP2 or 3-way subcutaneous, and that's all. You don't have to do any other vaccine. But this vaccine is extremely critical because I told you the class switching is not going to happen much faster if you're depending only on live vaccines. And to get that extra mileage of protection from 5 to 6 weeks onward till 9 weeks, you need the humoral support of humoral plus cellular immunity for the layers and the breeder, even the parents. And if you're going with the live vaccine alone, then day 5 to 6, the same 3-way or that IBD inactivated vaccine has to be done. And then check by deventer formula because layer chickens has higher half-life, IGY half-life. So it drops little late. So chances of subclinical Gumboro is much, much more in case of layer. And if at all Gumboro outbreak happen in layer, it will always have a late outbreak around 35, 40 plus days of age because of the extended curve of that half-life, the maternal antibody dropping because of increasing half-life. So normally you can do on day 15 and 25, 2 back-to-back vaccine within 10 days interval in drinking water. So in conclusion, IBDV is a resident virus of the farm and remains viable for a long period of time after an outbreak. So right biosecurity program is critical. So aldehyde-based compounds are effective against Gumboro. Avoid quaternary ammonium compound. It is not going to work, and detergents are not going to work. Even the bleaching powder and all, they have very slow action on Gumboro, and after chlorine, the available chlorine fades off, it has no efficacy. And selection of right vaccine strain is important. So if you're using conventional vaccine, that's live vaccine, And because it can displace the field virus over a period of time, and it can override the MDA and exert lower level of stress, bursal damage. So displacement of field virus from your farm means you have a farm with a vaccine virus in it. If it is genetically stable and it has a lower stress on the bursa, actually you avoid any field challenge for the future. So we have seen this doing IBDMB in India and many farms, which have been done in last 2 to 3 years. We don't see any outbreak. And when you do a PCR with the FTA card, we keep on doing this sampling from broiler farms randomly, where IBDMB live is done or MB1 is done. We find the burst even in flocks nearby which are not doing IBD/MB vaccines to have MB virus, the bursa. So it keeps on displacing the field virus. So that is the advantage. Selection of right vaccine strain is the key. Timing of vaccination is key to protection. Timing of vaccine virus entering the bursa. It's not the physical immune vaccination that you are doing in the chicks. Because your strain cannot override MDA, it will be delayed, it will be neutralized. Proper amount of virus not going into the bursa, so that cellular immunity will not kick in at right time. If proper cellular immunity is not kicking in, you don't have the humoral immune response later on. So your ELISA indicator will show poor uptake of the virus and may get the outbreak. And the hatchery-adapted vaccines are ideal for flocks having higher variation in MDA level, especially people who are not having their own parents and outsourcing chicks. Getting custom hatch chicks. So they don't have control on the MDA level. So they must go for the hatchery adapted vaccine. Even they can do vaccine at the farm level with subcutaneous injection between day 1 to day 3 of age. And I would suggest to use that first 1 week vaccination. I mean, day 1 to day 7. People using the Gumboro live vaccine in case of layers. Broiler, it's still okay. In case of layers, people doing day 1 to day 7 live vaccine standard, use it cautiously, because that is going to affect if you're going in the mucosal route. The hatchery-adapted vaccines are engineered in a way that they don't interfere with the migration process. But the conventional live vaccines that you are doing, it will have an implications in the chickens having lower MDA, because it might interfere with the migration process of the mature B cells to the secondary lymphoid organ. So that's all from today's webinar. We had a trouble in between. I had to skip some of the slides, but hopefully I can have some quick question answer rounds. Thank you.
Okay, am I audible?
Yeah, you are audible.
Thank you very much, Dr. Susi Mukulre, for your outstanding and such an eye-opening presentation. And it's been a matter of great pride and pleasure for us that we've been able to connect to you regularly for such technical talk. It's really been a matter of great pleasure for us. And in today's session, I would like to thank all the participants who waited so patiently during the disconnection, and sorry for the technical disconnection. And I can see some questions, Dr. Susim. There is a question from Dr. Rohit Reddy, and his question is, what is the difference between IBD intermediate vaccines and IBD plus vaccines?
So already, I think I have talked about this. The intermediate strains are more attenuated than the intermediate plus. The intermediate strains cannot override the MDA above 125. Suppose the MDA is dropping to 125, then you can do an intermediate vaccine. If you're not waiting till the MDA drops till 125, you are doing early, little bit early, the vaccine virus will get neutralized. Third is it has much lower stress on the bird due to its poor replication characteristics. The intermediate plus have higher replication characteristics. It have more immunogenicity, more replication, and it is recognized by the immune system much faster. So it has better breakthrough titer that we call for the intermediate plus, that is around 500 to 800. So this is the basic difference. And due to stress on the bursa, it will have a lower B by B index. But these are all transient damage. Even the intermediate plus, if you're using it cautiously, it can give you very good results.
Okay, there is a second question from Dr. Zay. What if we give IBD vaccine on outbreak of Gumboro? Can we do that?
This is very tricky questions. See, it depends on the size of the farm, your position of the shade. If you have not done right vaccine with the right vaccine strategy, Gumboro vaccination failure is also one of the biggest cause. We need to understand that Gumboro vaccination failure itself can cause an outbreak. It is not that you have timed the vaccination poorly. Of course, it is a cause. So if you are sure that the vaccine that you have done previously might have not worked, and you have got an outbreak in one shade and another shade is still not showing any sign and symptom, you may be quick to do another live vaccination in that particular shade. But in that shade where the outbreak has, the symptom has already presented, you might increase the stress by doing the second live vaccine, because the second live vaccine that you will be doing in that shade, definitely it's going to have a stress on the overall, because Gumboro vaccine is always a stress of the bursa as well as the immune system as well as the overall physiology of the bird. So avoid that. Because this, in my talk, I didn't cover about the pathogenesis properly, because I just focused on immunology, that after mortality, there will be surviving birds, and the B cells that are being destroyed, they will regenerate back or they won't regenerate into permanent immune suppression. But how long this disease lasts, that is around 4 to 7 days. It will automatically subside. So we try to give good immunomodulator, electrolytes, because there's a lot of dehydration. Because it's just inflammation, no? Everyone, we have a fever during this COVID or influenza. If you take a human as an example, if you have a high fever, we tend to have a lot of sweating, and at night, suppose you are having fever, you will get very thirsty, right? So this is a dehydration. So you need to check the dehydration, give a lot of electrolytes, that will help. But if it is in a different shade altogether, you can vaccinate the flock.
Okay, there is another question from Ganeshji. Since I couldn't understand the question properly, I request Ganeshji to unmute his microphone and put his question by voice. Ganeshji, please. Ganeshji. Okay, I will try to read the question myself. What is the breakthrough titer for MB1 in immunocomplex vaccine? And what is...
I understood. MB1 doesn't require any breakthrough titer because it's engineered to work individually according to the release. We consider it to be around 500, because breakthrough titer is considered for conventional live vaccines while calculating the right age of vaccination through deventer formula. MB1 is a hatchery vaccine that has been engineered in a way that you give in subcutaneous route, it will automatically adjust the release or delay the release of the vaccine in the bursa once the MDA level has went off. So you don't have to calculate or understand about its breakthrough. But still, answering your question, it will be around 500.
Okay, I can see a question from Dr. Prakash. Can you please unmute your microphone and put your question by your voice? Okay, he seems to be disconnected. I will put the question. Could you please elaborate pros and cons of hatchery vaccine, having both ND and IBD together and singly?
Yeah. Yeah, that is a very relevant question. I was talking about the vectored vaccine. So, hatchery adapted vaccine for ND has to be essentially a vectored vaccine, where the vector is going to be the herpes virus of Turkey. Now, herpes virus of Turkey is a slow replicating virus. It is just like commuting by a bicycle and commuting by a bike. So, but it is very steady. For Gumboro, we require early protection. So, and the virus and the disease is not relevant after 6 weeks or 7 weeks of age. For layers, maybe 8-9 weeks of age. So, the potential problem with slow replication with herpes virus of Turkey has a lot of disadvantages when you use IBD vectored vaccine. But for ND, the things are different. In ND, the cellular immunity also very important along with humoral immunity. And case of layers and parents, you need to do a lot of repeated ND live vaccines later stages. So if you do a vectored ND vaccine expressing the fusion protein along with Gumboro VP2, it's worth doing that. But and you can eliminate some of the live vaccines at later stages. And how many you can eliminate depends on a lot of studies that has to be done, depending on the data that has to be done by the company. As of now, we don't have any data, but definitely I'm sure by the replication status of HBT, because it's a lifelong carrier, the HBT will be there in the virus lifelong. It will keep on replicating. Again, it will go to the latency. If you understand herpes virus of Turkey, it is a herpes virus. So herpes virus typically has 2 stages of life cycle. One is latency, one is productive life cycle. In latency, it will go to sleep in the nerve ganglia, and with some stress, some triggering factor, it will again come back to replication. So in this manner, it keep on doing that throughout the life cycle, right? If you have a fusion protein expressing herpes virus of Turkey, that is ND vector vaccine, it will keep on expressing that fusion protein for lifelong. And it will keep on producing antibodies. Immune system will keep on producing antibodies against that fusion protein. So you have a sustained cellular immunity, sustained humoral immunity. But you can reduce some live vaccines in future. But the kill vaccines has to be done because it's only the fusion protein that is being expressed. You need other HN protein also, very important protein for ND, for protection. You need to do some live vaccines because again, the HN protein is missing. It's a very important protein for attachment. If you can block the attachment of the virus, then fusion protein is redundant. Last time I had a talk on Newcastle disease, so it's very important to have a live and kill vaccine combination, but you can definitely reduce some of the live vaccines in future. But the word of caution is, what about the first 4 weeks of the chick's life in case of ND, in case of Gumboro? So there are research publications. You can go through that, available in Google. You can type on the ND vectored vaccines limitations. There have been many researchers around the globe who have reviewed this, that if you're using a vector vaccine, maybe ND or Gumboro, it has to be combined with conventional live, because that conventional live will ensure that you cover up the first 4 weeks window period that is actually lacking with the vector vaccine. So that window period will be closed with the conventional live vaccines. So in fact, some of the manufacturer of ND vector vaccines, they recommend using inactivated and live ND vaccines along with their vector vaccines in their protocol. So in endemic country, you have to do it to protect the chicks for first 4 weeks or 5 weeks of their life. Hope that answers the question.
Okay, thank you. A couple of more questions. is it true that oral route of IBD live vaccination is more effective than the ocular route? And the reason behind?
Studies have been done. Studies have been done with both the route. There is no significant difference in protection. I must tell you that. It depends on the interactions, because when studies are done, it is only the Gumboro that is being studied in context of Gumboro. In the field, we are doing, suppose for example, in broilers, say, we do, I personally recommend doing a live vaccine of ND on day 1, another live vaccine of ND, the first one obviously in eye drop group, another live vaccine of ND on day 9 or day 10, because the MDA levels of the NDs are also very high. And day 1 in the endemic period, we also do combined inactivated plus live ND. So when you do the repeat live vaccine on day 9 or day 10 in eye drop route, and again you are going to do a Gumboro vaccine on day 12 in broiler in eye drop route, there could be some interactions and an interference. And that is why I prefer to use the drinking water route. And there you might get a difference in efficacy or protection level. But if you're doing a solo experiment with Gumboro, one in eye route, eye drop route, and drinking water route, there is no significant difference, although it has been shown that in eye drop route, in solo experiments, the antigen presentation is faster than the drinking water route. The reason being that the Hadarian gland is there in the eye, and the dendritic cells are able to present it much faster, be to quicker access. In oral route, it has to gain access to the gut-associated lymphoid tissues, which requires some bit of traveling, right? So that is the only thing that comes in the way. But in my experience, doing drinking water vaccination in such cases, where you do a ND vaccine just 2 days before eye drop route, it is much more, scientifically correct, and would be effective in this context in drinking water. And I didn't see any potential problems during the past few years with Gumboro being done in drinking water. All right.
Okay, I can see one more question from Dr. Prakash. I request Dr. Prakash to unmute his microphone and put his question by voice.
Thank you, Dr. Vivek and Dr. Susim. My question is, while doing practice in field, I found difficulty in diagnosing, differentiating IBD, heat stress, and IBS in broilers, especially in 6 to 8 weeks of age. So, if there is any clear-cut idea by postmortem examination, it would be helpful.
IBD and IBH.
Yeah.
You meant to say IBD and IBH?
Yes, yes.
First of all, it's very, I don't know how deep you have gone through. It's very gross. Gumboro speculated farm, you should cut open the bursa at the very, very first place. You will find hemorrhages, caseous exudate in the bursa. But I have a lot of pictures. That's another thing. Second, you will find thigh muscle hemorrhage. And these are 3 important pathognomic lesions I'm talking about Gumboro. You will find thigh muscle hemorrhage. And the confirming, a lesion that is confirming of Gumboro along with bursal hemorrhages, there will be hemorrhage in the junction of preventriculus and gizzard area. Although recent times we are not seeing with the vaccine being failed, where the vaccines are not able to provide proper protection, where, for example, say in 1,000 birds, where vaccine is not able to provide protection, I mean to say the vaccine uptake was not proper, the application was not proper maybe. So what happened, like say 100 birds or 200 birds were potentially unvaccinated and they were susceptible and they got the infection, but they were protected by the other fellow mates in the in the farm. This is called hard immunity. It creates a boundary and the virus to spread. But eventually, at later stage, the virus were able to amplify and increase their population to a certain extent, and they were able to eventually infect the flock, and the 100 of the birds died. So in such cases, you may not see the hemorrhage in the proventriculus and gizzard junction, but definitely there will be bursa hemorrhage. There is no other way. A Gumboro virus has to infect a flock by growing in the bursa. And once they grow in the bursa, if it's a field virus, maybe it's a lower pathotype also, there has to be a hemorrhage. Because I explained the IgM positive B cells is the primary site of replications for this virus. So there is no other way to go. So, and even the spleens are not involved, which you can see even in the IBH. You may see that spleen is responding to that infection, and it has grown in size, but the spleen's germinal centers are considered to be resistant to Gumboro replication. In IBH, it is straightforward that you get livers are involved, and livers are big, and you get kidneys involved. You get accumulation of water, the heart, even the peritoneum cavity. The leachy disease that we see, hydropericardium, it's now getting much more common. I mean, it is always there invariably. The liver involvement you won't see in Gumboro, and bursa involvement you won't see in Gumboro. But again, if it is Gumboro aggravated IVH, you might see that. But before that, you will see a lot of mortality happening before the IBH takes over, because IBH will start somewhere from 24-25 days of age, but it may start early, some rare occurrence from 16-17 days also, but normally it starts from 24-25 days of age, and adult birds liver are affected. And if it is aggravated by subclinical IBD, then it is more. And in subclinical IBD, you won't see frank hemorrhages in the bursa. The bursa tend to get hypertrophied, but you won't see that every rosettes and pillar of bursa follicle are getting red appearance. It's very pathognomic, right? You cannot differentiate subclinical IBD visibly. It's very unfortunate. Subclinical IBD, you need to do histopathology of bursa. You need to see the partial B cell depletion if you're really interested in. Or you can do a Gumboro titer, measure Gumboro titer at around 35.
Okay, I can see a question from Dr. Dipendra Kaundel. Dr. Kaundel, you may put your question by voice. Okay, I'll read his question. Do you recommend to give IBD vaccination by spray method?
Why would you spray? It's big. Why would you spray a Gumboro vaccine? Spraying is always try to mimic the natural route of entry. Gumboro virus do not transmit by fomites. Primarily, everything I should say primarily, because accidentally many things happens. If a farm has an outbreak and the secretion, even the droppings shedding, the virus shedded by droppings, it may turn into fomite and infect another bird. But primarily, fecal-oral route is a transmission for the disease. So spraying is something mimicking with the respiratory infections caused by the like say Newcastle disease or IB. So spraying is relevant method of vaccination for those diseases. Here you have a fecal-oral route of transmission. So better to give either in the eye drop route, because through eye drop route, remember it is going actually through some partly it is getting presented by the follicular dendrocyte in the local lymph node, but from eyes it again goes into the oropharynx and again into the intestinal tract. You got my point? So potentially it from respiratory tract also it is going from eyes also. The virus may have a fecal-oral transmission, right? So and an advantage is another, the Hadrian gland is present, so faster presentation. So I don't recommend doing any spray for Gumboro. It's relevant for IB and Newcastle disease.
Okay, thank you very much, Dr. Ray. It's been, we are running out of time. It's been more than 2 hours now. So lastly, I again like to thank the participants who waited so patiently during the disconnection. And also Dr. Susim, we would like to thank you very much for your precious time, for your valuable time. Despite the long disconnection and gap, you again came back and delivered such an outstanding and eye-opening presentation. And now I request to the coordinator of today's program, Dr. Yuvraj Pant, the secretary of NBA Lumini province, to take this forum.
Okay, thank you, Dr. Vivek, for moderating the program. I would like to thank Dr. Susim for his insightful presentation and giving us a very good information about the Gumboro and how to optimize the immunity level, because it's a virus-related disease and it has a high economic importance in poultry. So I hope every of us are well acquainted with the information provided. So I think we are a bit out of time, so I will be joining towards the end of the event. I'd like to thank again Dr. Susim. I would like to thank Dr. Vivek for helping me organize this program, and most of the events or most of the activities were due to him that we have come up to this. I would like to thank Dr. Krishna Kafle for being present here as a team member from Nepal Veterinary Association. So I hope the program was fruitful, and I hope the veterinarians or the related stakeholders who are present here have taken some information and they will apply the knowledge they have gained here. At the last, I would like to ask Dr. Prasant Acharya, who is the chairman of this event and the president of NBA Lumini, to present his vote of thanks as well as closing remarks, and I will be thank you.
धन्यवाद युवराज। आदरणीय गुरु डाक्टर कृष्ण काफ्ले सर, उपाध्यक्ष नेपाल भेटनरी एसोसिएसन लगायत यो जुम प्लेटफर्ममा आउनुभएका सम्पूर्ण आदरणीय अग्रजहरू, साथीहरू, भाइ बहिनीहरू ले हामीलाई चाहिँ आज धेरै कुरा सिकाउनु भएको छ। यसको लागि सबैप्रति म आभार व्यक्त गर्छु। Especially I'd like to thank Dr. Ray to providing valuable knowledge and information regarding IBD. That's why we would like to thanks from the Nepal Veterinary Association Lumbini Province as well as Nepal Veterinary Association Central Committee. यति कुरा राख्दै आज हामी समय पनि अलिकति बढी भइरहेछ। 1 घण्टा भनेको थियौं, 2 घण्टा समय दिनुभयो। सबैको लागि सबैलाई धन्यवाद। धैर्यतापूर्वक सुनिदिनु भयो। ज्ञान गुणका कुराहरू बाँड्ने जान्ने मौका पाइयो। विवेकले कोअर्डिनेसन गरेकोमा विवेकलाई धन्यवाद। युवराजले यो क्रियाकलापहरूलाई सबैलाई मध्यस्थ गरेकोमा युवराजलाई पनि धन्यवाद। लुम्बिनी प्रदेशको सम्पूर्ण टिमलाई र सबैलाई मैले चाहिँ धन्यवाद गर्दै अब चाहिँ हामी बिदा हुन्छौं। ल अब के गरौँ युवराज जी?
हजुरलाई प्लेटफर्म दिइसकेको छ र सकियो अब।
ल अब मैले पनि बिदा मागे। हस्, नमस्कार, जय पशुपति।
थ्यांक यू, बाय बाय ऑल।